On June 20th, at the 12th International Conference on Clinical Spectroscopy, SPEC2022 in Dublin, 4 special workshops were held. Together they explored the capabilities of the new Mirage LS and the part it played in recent cutting-edge research. This is a presentation of those 4 workshops. Use the bar below to navigate to any specific workshop.
All right, well, thank you everybody. It's a pretty good turnout. In case anyone doesn't know me, I'm Mustafa Kansiz. I work now for Photothermal Spectroscopy Corp. Some of you might remember me from my previous life. It's been 4 years since our last SPEC2020 conference, or SPEC conference, and unfortunately Mike missed out. And I think I did a workshop the last time around as well. So always great to be back. A lot of familiar faces, a lot of new faces. Yeah, so I'm just jump straight into it. We've got 3 speakers actually, so I'll be the first sort of introductory speaker. I'll give you a background on how the technique works and all the advantages, and then a few application examples, and then I'll pass over to the 3 of our guest speakers. So today I'll be talking about a new evolution in the instrument. So we've been around now for a few years. Optical photothermal infrared, hopefully you've heard about it. I think you'll hear a lot about it throughout this conference. But we're now moving on to the next generation. We're now combining, making a multimodal, trimodal, and we're taking advantage of all of the correlative approaches that are now coming out as well. So I'll introduce you, we'll go through some of the advantages and just advantages relative to FTIR. I'll go through how the technique itself works, some of the fundamental principles at a fairly high level because we're short on time. I'll introduce some of these new modes, new possibilities, and then I'll take you through a few. So most of you here, of course, are all vibrational spectroscopists. Could be infrared, could be Raman, could be a bit of both. When it comes to infrared, we all know we've got spatial resolution limits. It's fundamental, we can't do much about that. Sample prep can be a challenge for infrared. Thin sections are required. Some things you can cut, tissue's not so bad, but some things you can't cut. For that's an issue. For some of those, we can get around or work around that using ATR, but that's a contact technique. It can damage the crystal, damage the sample, and getting precise location can be a challenge. There's an example of indentations that an ATR tip can provide. When you work in biological, of course, you want to avoid touching it, don't you? One of the biggest issues, I think most of you can probably relate to it, are these dispersive scattering artifacts, mean scattering. And that can really impact the quality of the spectrum we produce through the spectrum. And here I've given an example of a thin film of a polymer. It's an ideal spectrum. Nice flat baseline, nice symmetric peaks. But if you take the same material and make it into a bead, spherical bead, in this case, it's 15 microns, the spectrum look very different. And change the shape of the bead, keeping the chemistry the same, the spectra change drastically. It's the same chemistry, just shape and size are now different. It really goes to show that when you work with these direct infrared techniques, your spectra can be highly morphology dependent. Now, I've used the term the direct infrared techniques. I've got another slide on this on its own, but I'll verbally explain what I mean by that. So with FTIR and some of these emerging QCell microscope techniques that are coming on the market, you've got infrared coming in, infrared interacts with the sample, and you measure the infrared that comes out. It reflects or transmits with an infrared detector, so it's direct infrared all the way through. Direct might sound great, but with direct techniques, you have these wavelength dispersive scattering artifacts. And what we do, and I'll explain that in a minute as well, with OPTIR is we remove all of these dispersing scattering artifacts. When it comes to Raman, most of you are Raman, it's probably less IR people here, but autofluorescence is the #1 enemy. Of course, we can go longer wavelengths, but you take massive heat and sensitivity. That's on top of the already limited sensitivity. There's photo damage, phototoxicity to living samples. So a few issues there as well. So with OPTIR, we've kind of overcome virtually all of those. And so, but what is it? How does it work? We can describe it as a pump probe optical spectroscopy technique. The pump is the infrared laser, and the probe is a visible laser. And that's kind of what I meant by the direct indirect. So we excite with infrared, and we measure indirectly with a visible laser. Single frequency Raman grade visible laser. Right, and what that means and what it delivers us is, among many other things, submicron infrared spatial resolution. Right, so it's much like a Raman microscope in that sense. Now, I'll make that comparison to Raman quite a lot. It generates FTIR-like spectral quality, even though it works primarily in reflection mode. We do have some transmission examples, but unlike regular infrared, where if you operate in reflection mode, the spectra look like crap. Here, you work in reflection mode, which is the easy mode. The spectra look like they've been collected with an ATR, or transmission mode. And it's non-contact, and the spatial resolution is wavelength independent. So whether you're at the low wave number end or the high wave number end, the spatial resolution is the same. Again, much like Raman. Right, so how does it work? Well, we start with our infrared laser focusing through a Cassegrain objective, a reflective Cassegrain objective, and that focuses onto the sample, being long wavelength, give a large spot size, really large. At the same time, we introduce collinearly a visible laser, typically a 532, can be 75, or another laser on request. And now with the short wavelength laser, we can focus it nice and tight, half a micron or thereabouts, and that's really where the bulk of that spatial resolution comes from. Okay. So then what we do then, as we're sweeping the infrared wavelengths from one end of the spectrum to the other, remember this is a broadly tunable QCell laser that we're using. As we're sweeping the wavelengths, we hit a resonance, an excitement at a particular wavelength. And that causes localized heating, that causes slight expansion, which will cause a change in refractive index. Right, so we end up synchronously monitoring the green laser. So we monitor the green reflectivity as we're changing the infrared wavelengths. And it's that reflected green intensity from which we extract a pure absorbent spectrum out of it. And then because it's a QCell laser, we can actually stop and park that infrared laser at any sort of given wave number, and then do single frequency imaging without any of the scattering artifacts that can trouble other single frequency approaches. Okay. So some new developments, and new by new is a bit relative, it's been around now for over a year or so. So our standard QCell is 1800 to about 950, and that's a 3-chip device. These QCells are made up of multiple chips, up to 4. We can also offer a high wave number OPTIR laser covering the OH and HCH stretch region. But my favorite, if one wants to have a favorite QCL, would be this CH fingerprint QCL, I call it. The one that covers the CH stretch and the fingerprint in one device. So that's pretty new, and you won't find too many QCLs that do the CH region as well. I know some people, Cassio for example, has a silent region chip. So instead of having this CH, we replace that with a silent region, and now so you can do your CD labeling, nitriles, and so on, any sort of triple bonds as well. Or if you really want to go down to the longer wavelength, the shorter wave numbers, we can have a wide range QCL as well. The probe laser is typically a 532, and we also a 785, and with the newer model, I'll talk about a little bit later, we can actually mount them simultaneously as well. Okay. People often ask me, well, how similar is an OPTIR spectrum and how comparable is it to an FTIR spectrum? Well, very. Here's where I'm going to try and show that to you. So here I'm going to show you polymer spectra that have been collected in transmission mode and a thin film. I'm going to compare that with OPTIR spectra collected in reflection mode of a thick block. Okay, so the first example here is polypropylene. Black, we've got OPTIR. In red, we've got the FTIR reference. You can see that's a virtually perfect match. And the spectra here are all uncorrected. That's basically raw spectra being spat out of the instrument. And for all the other polymers, the spectral equivalence is pretty much spot on. Right? So you need not worry about previous FTIR results not being comparable to OPTIR results. They're completely and fully comparable. Right. So at the moment, now I'm now going to move into the phase where I talk about some of these new capabilities. Right, so currently, or maybe let's call it the older generation instrument, we operate in what we call co-propagating mode. That's where the infrared comes from the top and the green comes from the top. That forces us to use the Cassegrain objective because they're very different wavelengths. We work in reflection, we can do transmission. As I said, we find a reflective objective. But what we've now done with the new model is have the option to bring in the infrared from the bottom. So that has some advantages. Maybe it's one negative as well, and I'll be perfectly honest about that as well. So we've now added the option of counter-propagating. And because the infrared now comes from the bottom, it requires a thin infrared substrate. So we're recommending a 350 micron thin fluoride window as the ideal substrate, so you can be a bit more flexible with that. And when you bring the infrared from the bottom, it really frees up your top objective, which is your collection objective, to be a high quality refractive glass objective. And then you can do lots of wonderful things with that you can't do with the Cassio Lima. That's what I'll show in the next few slides. And we've also added a fluorescence module, and that gives you that tri-modality. We're already bi-modal, infrared and Raman, and now we've got fluorescence and top. Right. So, as I said, infrared from the bottom, thin substrate, some sort of cellular tissue on top. What that means now, you can use really high quality objectives, 100x, you could use oil immersion, water dipping objectives, long working distance objectives. It really opens up the world of possibilities. Right? It gives you better spatial resolution, gives you better signal noise. These glass objectives have a lot more throughput, and just better imaging overall. And so, so in that mode, I've got a dry objective in the schematic. I'm now switching over to what it would look like with a water dipping objective. So you could use water dipping objective in the infrared. Sounds pretty strange and amazing to me. I'll show you the result of that. You can flip the sample upside down and work off a glass cover slip, sorry, a thin glass cover slip, and do oil immersion as well. Or you can work or image through a thick 1 millimeter thick glass slide with a long working distance as well. So I mentioned fluorescence. So we're kind of keen on addressing or pursuing this fluorescence space because, like all of you here, we're keen on addressing the life scientist. The life scientist has already used fluorescence for decades, several decades. So it's a workhorse tool. But now we want to bring infrared spectroscopy, and I do but primarily infrared, as an add-on to what they already do. And that's I think the key thing is to not force them to learn something new, but to give them something extra that they don't already have. And this is how we think we can do this. We can do co-located fluorescence and OPTIR. I'll give you some examples of that in a minute as well. But jumping around a little bit, some other examples of working in this sort of dry, high NA mode. These are some bacterial spores image. These are about 1 or 2 microns. It's pretty easy. This is a pretty astonishing result as far as I'm concerned. So this is a cheek cell on a glass on a fluoride, a drop of water, imaged with a water dipping objective, infrared from underneath. The green and the red are on the cell. You can just sort of make out the cell outline there. So the red and the green, you have cellular spectra, not water spectra. If you're out in the water, you have a bit of water spectra. Right? And then, of course, you can do single frequency and so on. Right? So again, there's no water correction here. So you can now do infrared and Raman of potentially live cells, right? with very little water background. Just for fun, because we're a crazy bunch in the lab, we threw in some 2 micron polystyrene spheres together with that, just for some extra contrast. We can see, again, good looking cellular spectra, even though we're in water, right? Water dipping objective. And then when you're on the spheres, you're getting polystyrene signatures. This is a study result that Kathy will recognise, courtesy of Kathy. You are there, last minute addition. And then, so this is oil immersion. So this is about as high a spatial resolution as you can possibly get. NA on these oil immersion objectives, I think it's 1.3, 1.4. So if we flip that upside down on a glass cover slip, imaged for the lipid, and I think this was some sort of cheek cell or a buccal cell. And so the spectrum from a point over there shows us lipid, protein, and a broad glass background. We are on glass. That's potentially correctable and subtractable. Yes, so it's 1.3 NA. Again, the spectra collected in seconds, image collected in minutes. And if I zoom in a little bit, you can see lipid inclusions of only a couple of hundred meters in size. So that's really pushing the absolute limit. We are working in infrared. This is far field. It's not a near field technique, which means you have all of the flexibility and the ease of use that you get with an optical microscope. Okay. Some more examples now when it comes to fluorescence guided. This is now our big bet, our big play to get into that life science community. So ease them into spectroscopy, not make them think it's something to be afraid of, but something that will give them some added value. Right, so this was actually collected on our microscope with that fluorescence module added on top. Now, when you have this fluorescence on the same microscope, it also means you don't have to worry about registration anymore. taking the sample from one microscope to a different microscope. Infrared Raman fluorescence means it's a registration nightmare. Here, it's all automatically and fully inherently registered. Protein image. Now, the protein image marries up with the nucleic acid imaging. It doesn't quite make sense, but it does make sense because it's quite dense in protein. But again, we're pushing this alignment to traditional life science workflows. We want this to be a complementary value-added technique to existing microscopy users. Right. I want to throw in a little bit here of microplastics because everyone hears about it. I know some of you, even though you're bio people, are probably working on it. One cool approach is that you can actually tag your microplastics or other components fluorescently. You can then image for that through the fluorescence channel, and that's exactly an example of polymer beads that have been fluorescently labeled and imaged on our platform. If I overlay that with the visible image, you see that these line up with the particles, but there's also a lot of junk in there. A lot of junk in there that's not actually polymeric. So without the fluorescence channel, you'd be off there measuring all those other particles and only maybe half of them are polymeric. Whereas now you know which ones are polymeric and you go only to that. So you can drive the system based on the fluorescence image and or the visible image. Right? And because the fluorescent tags are actually such low concentrations, they don't turn up in the visible in the spectral channel. Okay, some real world sort of bio, these are, I think these are grain tissues. These have been labeled with a marker for beta amyloids. And you can see where they light up, but some of them aren't actually visible in the visible channel. Right? You can only see them in the fluorescence channel. And if we take a line scan across that and average for the blue, which is on the beta amyloid, and red off, you can see strong, clear, and expected differences spectrally with protein secondary structure. And this one's a very new data set from about a week or two ago. So I've forgotten what type of cell this is, but just to show you how visible and the fluorescence and the infrared work together. We've got nucleus in blue, the lipids in green, and we've got stress granules. That's a new term for me, but I didn't know if they were stress granules, but they are. And of course, when you take a spectrum on the green spot, lipid, you get a copy great lipid signature. When you're on the blue, you get some strong nucleic acid features. I think the next spectrum, I have an example here of spectrum on the stress granule. The stress granule is a little bit different, a little bit shifted, would be indicative of some protein secondary structure differences. And these are all brand new data sets, and this is on an unpurged system as well. So basically an open cover means we see a little bit of water vector through. This here is a 500 nanometer sphere, just to give you a sense of the resolution. You can see we can easily resolve a 500 nanometer sphere. And the spectrum from that is also straightforward. Probably should have done this earlier on, but infrared and Raman, that's kind of our central value proposition. I call this the holy grail of vibrational spectroscopy. Same spot, same time, same resolution. How does it work? Remember the infrared beam? Bring that to our objective. We bring the visible probe at the same time. That's where the photothermal magic happens. And the reflected green light, or the transmitted green light could be as well, is sent onto a visible detector from which we extract our infrared signal. But because we're using a visible grade Raman laser, there's always Raman scattering going on, whether we like it or not, whether we want to use it or not. What we do, we use a little dichroic filter to separate out only the Raman scattered photons. We send that to a Raman spectrometer. And it's actually a very efficient way. People ask me, is there a compromise? Actually, no, not at all. You use virtually all of the Raman scattered photons, and the remainder, which by far the majority, goes on to visible detection. So here we get simultaneously, literally, infrared and Raman at the same time. Another example, so these are 500 nanometer polystyrene spheres. So a single bead, this is literally in seconds. So we've got in red the OPTIR, in green Raman. And my point here is that, whether you're dealing with a 500 nanometer sphere or a cluster of particles, the spectrum look the same. This would not happen with these direct infrared techniques, if you were FTIRs. You would get awful scattering phenomena going on. Here, the infrared looks the same. Again, not corrected. This is raw data coming off the instrument. And you can actually take advantage of the multiple, of the simultaneous search features of these library search engines as well. So you can, in fact, search infrared and Raman simultaneously as you're collecting them simultaneously. And in one axis, plot the Raman heat quality index. On the other axis, you plot the infrared heat quality index. So your upper right-hand corner is where your infrared and Raman matches really well. Gives it extra reassurance, that sort of confirmatory channel. I think I'm nearing the end. This is something, one of Cassio's samples that you've sent to me over the last year or so. So this is a single bacterium E. coli imaged in the infrared with 50 nanometer step sizes. The sampling significantly, of course, still relative to the green, but still you can see the clear E. coli shape. It's about 2 microns long and about a micron wide, and spectra collected. I think these are a few tens of seconds maybe, but the infrared in red and the Raman in green. So a single cell bacterial infrared is now quite routine. Right, I'm going to skip through those pretty quickly because I want to pass over to, I think, Peter, you're up next, I believe. So, yeah, thank you. I'll let
Thank you, Mustafa, and thank you for letting me speak today. So I'm really just going to continue the theme, and there's a little bit of overlap, but we were looking at OPTIR and concomitant Raman, single cell analysis. It's what we're interested in, so why are we studying single cells? For a wide range of reasons. Possibly infrared cytology. If we've got clinical samples, we can actually get quite a lot of information from them. We did a lot of work recently in stem cells and looking at stem cell differentiation, and that's quite important. We're looking at drug cell interactions, and much more recently doing a lot of work on drug delivery systems with AstraZeneca. Okay, so most of you will be very familiar with a biological cell spectrum, so I don't have to really go through it here, but we've got the fingerprint region, we've got proteins, phosphates, carbohydrates, etc. We've got lipids, we can get a fingerprint, but we do use the whole of the spectrum, and that is quite important. So I'll come on to that in a minute. Obviously, the advantage is the spectrum consists of all the constituencies. We get chemical information from within the cell. We're looking for particularly changes in cancerous cells, non-cancerous cells, etc. for a lot of the work we do, and metabolic modifications of those cells. So the disadvantage of conventional Raman, Mustafa has mentioned it, it's the spatial resolution, depending on how you do it, but it's a few microns to 10 microns, 12 microns. So we're also got to push towards live cell imaging because there can be fixation artifacts, etc. So the ultimate aim of what we want to do is live cell imaging with infrared spectroscopy to get subcellular information. So that's the direction that we want to go in. So that's what we're into. Mustafa has already shown this, the problem of the spatial resolution. And importantly, that the spatial resolution in an infrared, conventional infrared technique is wavelength dependent. And that can be a real problem. So if you're working at the high end of the spectrum, it might not be too bad, but if your information is relevant down here, suddenly your spatial resolution really deteriorates. So it's quite important to bear that in mind when you're analyzing images. We've been trying to get around this for a long time. We did some lovely work at the now, I think it's now obsolete, the Irene beamline in Wisconsin. And we got, there's some scattering artifacts in this data, really high pixel resolution, but ultimately we couldn't get really good subcellular resolution because it doesn't matter how many pixels you have, actually you can't get around the diffraction limit. So, you know, we were able to basically detect a nucleus and a cytoplasm, but we couldn't really get the information that we wanted. So we were very fortunate to get the first OPTIR system in the UK. I think it's one of the early systems. I won't go through how it works because Mustafa has already done it far better than I can do it. But the key thing I want to point out is that now because our spatial resolution is determined by the green laser, it's actually consistent across all the wavelengths. So that makes your image analysis a lot easier. And it's around about the half micron using the 532 laser. Okay, so it's not clicking again. What's going on? There we go. Okay, so on our particular system, we've got the QCL laser. I don't know what's happening to the pixelation on that image, but there's an image. It's a cell, and this is the QCL region covering the fingerprint region. But we can add the Raman spectrometer, which is really useful. So we've got the system with the Raman spectrometer, and you can see now beautifully the complementarity of these techniques in the fact that we've got really good information from the CH stretching region, but the AMI1 and AMI2 is quite low in Raman, quite low intensity, whereas of course it's high in the infrared. But we're also lucky enough to have the OPO system as well. So basically, we're now in the fortunate position where we've got the QCL that delivers the fingerprint region, we've got the Raman that goes right away across the spectrum, and we've got the OPO that gets us the high wave number region. So in a single instrument at the same time on the same spot, we can collect data across the whole spectrum, and we can do a much more complete analysis of our data. So the aim of our studies was to use the OPTIR system to identify subcellular structures in fixed and live human cancer cells. So this is the first study that we did with this instrument. The concomitant Raman is very important to us, same location, same time, along with the QCL and OPO laser excitation. And we wanted to develop some sample preparation methodologies to optimize the technique. And originally we had a very simple system, just using 2 calcium fluoride plates where we could just put the cells on. We used a pack pen, just put them together. The pack pen, which is gives you essentially a water-resistant barrier, was enough to just give you a few microns gap for the cells to be in, and we got a very good data. I'll come on to that. Okay, let's show you some results. It works beautifully, I have to say. So here is the optical image of a cell. We would have loved to have had a fluorescence module, but it wasn't available to us. But here is the fluorescent image. It's not the same cell. And this is important because it backs up Mustafa's issue with trying to do registration, etc. So, however, these are Mia-Packer cells, which is a pancreatic cancer cell. But basically, we found one that looked almost exactly the same in the same orientation. And you can see this is actually stained for the nucleus here. So it's got a large nucleus, it's got a nucleoli here. And then this is actually the endoplasmic reticulum that has been stained. And you can see that there is going to be some match-up with this particular cell. What's interesting is, obviously, we can see within the cell very large differences in the intensity of the spectra and the relative intensity of the band. So we're now starting to see subcellular features that we couldn't see when we went to the synchrotron. So if you can just about see this light green spectrum here, we've got quite a strong protein signal. It's actually higher than your lipid signal here. But if you take this olive green spectrum, which is here, you can see we've got almost no protein here, but a certain but lipids. So we're starting to see the subcellular structure. Noticeable variations in the amide A and B. Let's say the olive green spectrum, it might be a lipid droplet, we're not sure. Variations in amide 1, amide 2, and we've got variations in the fingerprint region. So we know, you can see that we're seeing differences on length scales of fractions of microns. Okay, so if I actually normalize the spectra, actually this olive green spectrum jumps out now because it was low intensity elsewhere. But again, we can see really nice changes in intensity here. If you look at the green spectrum, you've got a complete switch of the ratio between the CH2 and CH3 compared with the olive green spectrum here. So that's the olive green spectrum, and that's the light green spectrum, which is actually in the nucleolus. So finally, we're starting to see that subcellular structure that we really wanted to identify. And we can also see it in the Raman, slightly different because the selection rules for Raman are slightly different, etc. But you can see there's a lot going on in the structure here in the Raman that actually confirms the infrared. So there's generally more variation in the infrared than the Raman, but it's compatible. Okay, I'm going to quickly skip through that because I'm conscious of time. Okay, so these are still fixed cells, and it's a different cell image, and this is now a sort of much more detailed scan again across a particular region. And look at the variations across this region. It varies quite significantly, and we've got the blue and the red spectra. I've got proteins and lipids in, but the olive green spectrum seems to be devoid of the protein, so it's mainly lipid, and we believe that this actually matches up with the endoplasmic reticulum. Again, I'll skip that, but we've got an endoplasmic reticulum, and we believe we were scanning over this area here. Okay. I'm going to just look at the AMI1 band, because obviously that's a very important band in the infrared. So this is the AMI1 band, and of course then we've got the CO stretching the carbonyl band here, and again very strong variations within the cell. If you look at the blue spectra, it's quite high, but if you look at the black spectrum, for example, it's completely disappeared. So if you look just within the nucleolus, or just going across here, I'll blow that up. The black, purple, and the green spectrum obviously have got no carbonyl band at all to speak of. Also, there is a broadening of the carbonyl band very significantly in the the AMI1 in the nucleolus. Okay. So there's some changes in conformations within the nucleolus. So it's always very risky looking for secondary structure with proteins in the infrared, but nevertheless, if you look at the second derivative, you do get very clear and very consistent data. You can see here these are perfectly consistent, and we get the same peaks every time. We were worried how much of this variation is reliable. Can we rely on that data? Turns out we absolutely can. I was quite surprised, and this is all completely reproducible, and it shows you that certain things are changing very quickly in the AMI1 band. So we can potentially assign it, tentative assignments, but it was a surprise to us that this was so consistent in our spectra with the OPTIR. Because one of the things when you first get a brand new instrument is you're a little skeptical. You think, okay, how well does it really work? But actually it does. So the Raman is very helpful. We've got the AMI1 and AMI2, but again, the complementarity, you can see the AMI2 virtually disappears. It's really helpful to have the Raman alongside. Look at the AMI3, and again, you can see, this won't be any surprise to anyone who does Raman spectroscopy of cells, but the beauty is you can combine it with the infrared and really confirm your assignments, etc. And you can see that there's huge differences here in the AMI3. The other thing that's really useful about the Raman for us was that we could get the much lower wave number spectra from the cells, and that helped because our OPTIR QCL goes down to about 900 wave numbers. But actually, when you look further down in the Raman, you can see that it looks a bit of a mess, but actually it's actually really consistent, and I'll show you why. There's broadly three bands, round about 770, 740, and 714, and you can see those within here. But if I just sort of deconstruct that a little bit, the intensity is very different. Here we have the DNA peaks, and that's from the green and black here. But if you move off that to look at the green and the blue here, you can see it's predominantly protein, complete switch, which is confirms exactly what we were able to tell using the OPTIR. So let me come on to the live cell work, because that's important. That's our ultimate aim. So the cells need to be in a buffer solution. We've got to keep them alive, obviously. The IR beam normally will not penetrate more than about 10 microns of water. We've done a lot of work at the synchrotron at Diamond, trying to develop techniques that will enable us to do the live cell imaging with a certain amount of success, but actually this is easier. We've done some work with a microfluidic cell that we will have been lent, but the work I'm going to present is actually from our original data. So this is some of the work at the Diamond synchrotron. Even with a short path length, what you can see is, sorry, that's the scale is the wrong way around, but you're dominated by the water spectrum, and you can see some cellular structure here, but you can't get anything in the CH stretching region, and you have to work hard to pull this out. So actually, no, I'm not going to show results from that. I'm going to show results from our original work, which is with using the PAC pen. So these are mere PACA2 cells, the pancreatic cells, cancerous pancreatic cells. These are breast cancer cells. We've done a lot of different cell lines. I'm not talking about the biology. I'm just trying to give you a feel for what we can do. So you can see this is just using the QCL in the fingerprint region. But again, I just want to point out the differences here that are very clearly defined on a subcellular level. And the red spectrum here is the water. So you can just see the buffer, but it's it means we're not getting the interference that we thought we might get, which is beautiful. You can see that the AMI1 and AMI2 ratios are just about what you would expect. And you can see this here as well. So the water contribution, once you go onto the cell, is minimal, which is fantastic, even though we've got a reasonable gap. We've got, let's say, the 1088 band, almost certainly the nucleolus in each case. We can see the change from when we hydrate the cells. It's different from the fixed cells that we had before because of this change in DNA conformation from A DNA to B. Sorry, A DNA to B DNA. Okay, let me skip through that. So I spoke a lot about the AMI1 vibration and about the consistency of the second derivative bands, because in the fixed cells, what happens when you go to the live cell, when you've got water that completely dominates that region of the spectrum, if you, in a normal experiment like the ones that we have done in our own lab or at the synchrotron. But when you do the second derivative, actually it's also perfectly consistent, slightly different, but again, I was absolutely amazed that we could pull out the information that we could pull out with the cells in water. the cells are still alive. We've done tests after all of the experiments. We can keep them alive for many hours. And this was a complete surprise to me that we would actually be able to at least see some structure and some structure changes. And the changes that we can see, depending on where we are in the cell, are completely consistent in the amide-1 and looking at the carbonyl region, completely consistent with what we saw in the fixed cells. So to be able to do this in water was a major breakthrough for us, and certainly was showed that this is the technique that we need. So you can actually assign these if you want to exactly how reliable it is. We're still doing lots of work on this, but nevertheless, the fact that it consistently comes out it wasn't random, and that's the key thing. So the OPO, this is again the live cells, which is really nice to have the OPO as well as this is the QCL. So this is just an image of 1550. And you can see we've got the bright spots, which I'm not going to go into. But when you have the OPO, it's exactly the same cells, practically exactly the same area. And you can see, obviously, because we've honed in on 2929, we're starting to see the lipids really light up. And we can, again, in our system, that's sort of about 1 micron. We can see things that are slightly smaller than that, so it's reasonably consistent with the 0.5 micron spatial resolution, which is pretty good. And this is in water, so that's the amazing thing that I have to keep telling myself. This is in water, so it's quite special. And also the bright spots in the lipids don't always give exactly the same spectrum, so there are changes even within those bright spots that they're not exactly the same. So we're seeing chemical variation on a scale that we couldn't previously see. Okay, I'm going to skip through that fairly quickly, but just to show you, again, the fluorescent image is not of exactly the same cell, so it would be lovely to have the fluorescence attachment in the new system, but what you can see is the spots that we're seeing are very consistent with the spots that we were seeing in the lipid image in these particular cells. So again, we've done the fluorescent imaging to try and always back up what the infrared is saying, and every time it seems to be pretty consistent. So we're very pleased with that. Okay, so that's just a quick overview of just a couple of cells of what we've been able to do with the system. So I think this was when we published this, the first use of the OPO, the QCL fingerprint, and the Raman for fixed and live biological cells. We've got high spatial resolution. You can see from those lipid images we were down to about the 0.5 micron in biological cells in water, which is the key point. So the IR and the Raman was done exactly the same time on the same spot, which is fantastic. So spectra collected a pure across the entire spectrum, so we haven't got the pixel mixing, we haven't got the problem of changing spatial resolution. We've got relatively undistorted amide-1 bands in live cells in water, which is amazing. So hopefully the implications that we can start doing more enhanced studies of cells, drug-cell interactions, and looking at cells' response to external stimuli, which is our next stage. And had the pandemic not come along, we would have had a lot more data by now. To conclude, really, the person who did most of the work is Alice Spedia, who is a brilliant Italian postdoc. Joe Denby was also involved in the project, Jane Lawrence, and Marion Ashford and Mark Jackman from AstraZeneca were central to helping us develop the OPTIR, and we've got some beautiful data on stuff that I can't talk about. Okay, on that note, I will say thank you very much indeed, and
Thank you. Thank you, Peter. But if there are any questions, we could time now, please.
When you are doing Raman, IR is always on. So your molecules are distributed in the So you think that stokes and anti-stokes will have equal intensity in that case?
Well, we don't measure the anti-Stokes. We're only measuring the Stokes vibration and the Stokes scattering. But I think it's pretty quick. I mean, it'd be an interesting experiment to do to see whether or not you start if you have if at some point when you're pumping you start to increase the vibrational hot bands and things like that. But that's sort of outside the scope of what we have done, and we don't measure the anti-Stokes, so but it's an interesting spectroscopy question.
Yeah, because if you can go that side, then at 5:52 fluorescence issues there for
Yeah, I mean, it might be something that I don't know whether you.
That question reminded me of something that I heard somebody suggest, and you know how with for Raman you can do visibly resonance enhanced Raman just by tuning the excitation to a particular visible excitation. You can do the same with vibrational as well. So you could, it'll be interesting experiment. I haven't done it, but if you were, if you had the QCL sitting at 1 band and always on, so you're always exciting at 1, and did the Raman on that versus having the QCL off or on a non-resonant band, might there be some Raman enhancement by virtue of there being an infrared?
I don't think you get a Raman enhancement.
Would it not be analogous to having resonance enhanced Raman?
No.
No, ignore me. It was a suggestion somebody made. I'll blame them.
I don't know. I don't think so. So basically, you're changing the population of states. Actually, your Raman spectrum might decrease because you've got fewer in the ground state. But yeah, we'd have to think that.
Okay. Any other questions? Okay.
I was wondering how the battery is controlling the lifestyle in the sensor. Is it because you showed a diagram, but then you said no, it's not this one?
Okay, so all of the results I showed, we had just a calcium fluoride disc. We used a pat pen to basically draw a barrier. We put the cells in, a drop of the cells that were in liquid, let them settle, let them attach, okay, and then slowly just put another calcium fluoride slide on top. So they are in water, but the pat pen gives you a big enough depth to actually keep the cells alive. And we can do, Alice is a cell biologist, and she's done all of the tests to make sure they're still alive. So it works. What we then wanted to do was to use a microfluidic system, and Mustafa very kindly lent us a microfluidic system, and we've got that, and we had some preliminary data with the microfluidic system. One of the problems we had was we've got some results from that, but we've been having issues with the cells for some reason not attaching properly, and then because you have to turn it upside down because you need the cells attached to the top surface. With the microfluidic system, we were having some issues with them not attaching properly, and we're not quite sure why. And when they're on the bottom surface, then you start to see a bigger water contribution. And so there's still some issues, but we've got a guy working on that. But, yeah.
So I want to stress something you said there. I call it the upside down sandwich. Because the infrared is coming from the top, having the cells on the top window hanging upside down is critical. The infrared will hit the cells first without having to travel through water first in the non-upside down direction. I think that's a big factor in minimizing water. I think another question.
Related to this answer, so what if the microscopy is written in the calcium fluoride, right? The channel.
It could be.
It's not in this case.
No, but it could be. I
In,
mean,
in your case, was in what? In glass?
No, it's so you need a calcium fluoride top plate, but it's machined, and you basically got a channel that goes into a gap between the two plates, such that it can flow through with it, and there's a spacer seal. But there's still some issues with the design that need to be worked out on that. So because machining we have machined calcium fluoride. It's really difficult because it's quite brittle. It's very difficult to do. So we built a microfluidic cell that we used at the synchrotron with machined calcium fluoride, the channels in calcium fluoride. But it's a really difficult process, so we need a different solution. And there are options to do different things. So.
Add to that as well. So you only need one window to the calcium fluoride, and the other window simply has to be visibly transparent. It could be plastic, it could be glass, it could be anything else. Only one window needs to be thin, infrared, and the rest can be. Okay.
I've got a spectroscopy question. I was trying to figure out, you said the endoplasmic reticulum, was that high in the kit or low in the kit?
High in the kit.
Oh, okay. Misheard.
Yeah, no, it's high. It's because it's got loads of lipid bilayers that do all of this, and the proteins are more transient visitors to the endoplasmic reticulum. So I think that's what we're seeing.
Gotcha.
About resonance Raman, so maybe already asked that. Anyway, I was wondering if matters you have in your sample Raman scatter or resonance Raman scatter. Out before, I don't know if there is any difference because I still don't understand really the physical evidence behind. Is any difference if some would rather in your sample resonance Raman spectrum or Raman?
I think.
There is an absorbance gap by the resonance Raman.
So I don't know. I don't know. Do you get much resonant Raman at 532?
Oh, yes. Yeah, proteins mainly.
Okay. So then that is going to, yeah, if there are things in there that are going to give you resonance Raman, you are going to get an the Raman is just like any other Raman process. You know, it's just like doing conventional Raman spectroscopy. You're putting a green laser in and you're measuring the scattered Raman photons. So if there are things that give you an enhanced Raman spectrum, it should jump out at you. Having said that, we haven't seen anything that looks out of the ordinary. So I'd be interested to know where we might find those things.
Yeah, you can see cytochromes with channel. Because in the resonance Raman, the thing is, for example, I work with bacteria, so they produce a lot of pigments that we can see due to the resonance Raman effect. So usually, for example, if you use 785, the spectrum is going to be dominated by proteins, lipids, carbohydrates. But then if you move to the lower, to 532, for example, then you will be able to amplify the Raman signatures from molecules that are usually low concentration that you don't see when you use 785. Like so cytochromes you can see, because I will show also on the next one. So I will show some Raman signatures from cytochromes. You can see pigments as well. So you can see both.
You see all of the Raman spectrum, resonance Raman spectrum. I was wondering if it affects somehow this photothermal spectrum actually simultaneous.
I don't think it does.
No.
Because the excitation is by the infrared, and the detection is the Rayleigh beam.
Yeah, you can, for example, turn it off the QCL and do just Raman. So you can turn it off the IR part.
I understand. Raman or resonance Raman. It's still wondering, but anyway, I will discuss it.
It's probably not a bad issue. I have a question from an online attendee. And the question is, I'll read that out. How can you confirm that the cells are alive/viable by just looking at the spectra and not apotopic, or not dehydrated, not stressed? And when you say water, do you mean cell media?
So they're in a buffer. They're in a buffer solution. So it's not cell media. They stay alive for. We have put it in the paper, and Alice has done various biological tests. I can't remember which one, but it was a trypan blue exclusion assay and a couple of others as well, because again, that was one of the things that we were really interested in, to make sure that the cells are alive. The issue of stress is a good one. I know that Lisa Vicari has published a number of papers on if you squash the cells between two plates, then there's some evidence that you are initiating some kind of stress response. We don't think our cells are particularly squashed, so we know there is a water gap. So I think we're okay, but we would still need to do. We couldn't completely rule that out. So there's a lot of work still to be done. It's a very good question, but I don't think we are stressing the cells too badly. When we have got spectra of the same cells in the microfluidic device, when the cells haven't fallen off, then they match quite quickly, and there's a slightly bigger gap in the microfluidic device compared with the sandwich that we use. So you know, I couldn't say definitively that the cells are not stressed at all, but I don't think we're seeing the same levels of stress that was in Lisa's study, where they had successively smaller and smaller gaps in the cells, so they were being threshed, and they see some slight changes in the spectra that they've attributed to a stress response. Although I still think there might be optical effects going on in there as well.
Our next speaker is the funny and entertaining Cassio. We'll see what I mean in a second.
I don't know what you're talking about. So hello everyone, nice to meet you all. Good afternoon. So before we start this presentation, we need to address something that I think is very important in science. Science is very international, which is something, you know, very nice, but sometimes we come from different cultures, we come from different countries, so our names can be a little bit challenging sometimes. So people complain about my name all the time, like Cassio, who has this name? So that's my name, and if you guys forget my name, just remember it's similar to a very famous brand of watches and calculators. I do have a sister, her name is not Rolex, I had this question before, okay? So I work at the University of Liverpool as a postdoc. I work with Professor Roy Baker. I would like to thank Mustafa. Is it Mustafa or Mustafa? Mustafa. You see, I've been saying his name wrong my whole life.
I've not given up.
Thanks for inviting us to share results. He actually invited Roy, but he wasn't available, and you guys got stuck with me. So now I'm going to drag you all to the microbiology world. So there's going to be a lot of bacteria here. So this is me in 2019. This is like my first week working in Liverpool. I am a physicist, so I knew nothing about bacteria, microbiology. I had no training in microbiology. I barely knew how to use a pipette. I still don't know how to use the multiple channel ones. I struggle with that. But the most important of all, so I was about to start working with bacteria. So this is a picture from my first week. This is the first time that I ever prepared a petri dish to grow bacteria. I was super happy. There's no contamination. Now I have to do this every week. A lot of times I'm not that excited anymore. But the problem was I was working with bacteria. And I was like, oh my God, I was holding the tubes like this because I was terrified. These are like biological weapons. I was obviously wearing gloves. We work in a biosafety cabinet, but it's still very dangerous. And I would be like, do not touch your face the whole time. Because bacteria obviously has a very bad reputation because they can cause diseases in humans. Nowadays, after 3 years working with bacteria, I have a different impression about these amazing microorganisms. There is a relatively small group of bacteria that can cause diseases in humans, but we do have a lot of bacteria in our body, in different organs, and these bacteria, they play an important role in the functioning of the human body. Sometimes these bacteria that should be on the place A, they move to the place B, and then they grow happy and fat, and then they cause disease. I don't know who that guy is. Just found that picture on Google, but he has a sty in his eye, which is a bacterial infection. Bacteria play an important role in the biogeochemical cycles, recycling nitrogen, recycling sulfur, recycling a lot of elements. We use bacteria quite a lot as microbial cell factories to produce a lot of metabolites that are important to us, like drugs, anti-cancer drugs, antibacterial, antifungal, vitamins, pigments, and my favorite, bacteria can also be used to make food. And so for those reasons, it is very important to study bacterial metabolism. And Raman and infrared spectroscopy, vibration spectroscopy, are great tools to study bacterial metabolism. So here we have the infrared and the Raman spectra collected from bacteria. The peaks that we see in the spectra are associated to the main macromolecules, like proteins, lipids, carbohydrates, nucleic acids, and products that are produced by bacteria, microbial products. So basically, both infrared and Raman spectroscopy provide information about the overall biochemistry of the samples. And for this reason, the two techniques are great options to be used in phenotyping studies. I think we all know that Raman and infrared spectroscopy work in completely different ways. So Raman is based on the scattering of light, infrared is based on the absorption of infrared radiation. And due to these different characteristics, there are some advantages and disadvantages on using them regarding the application to study bacterial metabolism. So it is quite common to use both techniques in the same study to get a better characterization of the spectroscopic features of the samples. And on these situations, so for OPTIR, we will have to prepare the samples twice because we will need infrared spectrometer to acquire the infrared data and a Raman spectrophotometer to acquire Raman spectra. I am very lazy. I hate pretty sample preparation. So with OPTIR, as you guys can see, we really enjoy the instrument and there is no need to prepare samples twice, which is something that I love it. I was a little bit overrating this picture. This was like after quarantine. So, you know, I like to say that this is my heavier isotope version of myself. Doing Raman and infrared simultaneously is not the only advantage of the OPTIR system. There is also the spatial resolution that Mustafa mentioned before, and Peter as well. So with the OPTIR system, we can reach 500 nanometers. So bacteria, they're tiny, tiny little. They're usually like 1 micron, between 500 nanometers and 1 micron. So if we use a standard FTIR spectrophotometer, we couldn't see single bacteria because they're too small. Obviously, before OPTIR, we could use IR snow and AFM-IR to get infrared signatures from bacteria, but they're a little bit time consuming. So basically with OPTIR, it combines the best of the two worlds. So we have crazy resolution to see single cells, bacterial cells, and it is quite quick. So this image here, it takes like 10 minutes to collect this single frequency image. So well, it is very important to understand what happens at single cell level in terms of bacterial studies, not only to understand the role and the function of the individual cells within the microbiome community, but also to understand the interaction between microorganisms. This will hopefully help us to understand the early steps in bacterial infections, to elucidate the spatial structure of communities, to pinpoint the emergence of pathogenic and antibiotic resistance, and to optimize the production of microbial products. As I mentioned before, we use bacteria quite a lot as factories to produce a lot of stuff to us. So now I'm going to share the results from 3 different studies using OPTIR spectroscopy. So in the first one, we use E. coli. So this is a standard bacteria. This is a friendly one. This is a friendly strain. So this one does not cause diseases. And we prepare the samples in 3 different ways. So the first experimental group, the cells were grew in regular glucose and regular ammonium chloride. So glucose is a source for carbon, ammonium chloride is a source for nitrogen that bacteria use to grow. And on the second group, we use labeled 13C glucose and regular ammonium chloride. And the last group, we use both labeled compounds, so 13C and 15N. We analyzed book populations using OPTIR and FTIR, and we also collected Raman signatures simultaneously to the infrared signatures using OPTIR system. And we also evaluated single cells using OPTIR. So here we have the spectra, the infrared signatures acquired from book populations using FTIR, book populations using OPTIR, and from single cells using OPTIR. We can see some shifts comparing the spectral signatures from the different experimental groups. So for example, so the amide1, which we usually see peaking around 1655 in unlabeled bacteria, when we grow the cells in a special media like 13C glucose, that peak shifts to 6016 because the molecular vibrations are affected by the incorporation of heavy isotopes. So this is a heavier atom. So it affects the way that the vibration works. So there is a high reproducibility in all three approaches. There is a lot of studies using FTIR spectroscopy to study bacterial samples. So first, we need to compare the OPTIR results, which is a new technology, which is with the standard technology, which is FTIR. So there is a high reproducibility in all three approaches. So here we have the PCA. The clustering patterns are very similar. We can discriminate all the three groups. So basically, there's no differences on results obtained by OPTIR spectroscopy compared to FTIR spectroscopy from book populations and single cells. So that means that the incorporation of heavy isotopes is very homogeneous at single cell level. So here we have the Raman results. Once again, we can see some shifts on the position of some peaks, which is also because they're affected by the incorporation of heavy isotopes. The clustering patterns are actually very similar to the clustering patterns that we obtained for infrared signatures. We did not perform one analysis from single cells because we were inducing sample degradation. So this is something that we need to be very careful with OPTIR because now we have two lasers illuminating the sample. So we have a QCL, we have a green laser, or we can have an OPO as well. And so here we collected 10 spectra and 50 spectra from single cells using OPTIR, from book populations using OPTIR, and from book populations using Raman, which was collected by OPTIR. We apply PCA, and as we can see on 50 spectra, there is a trend. So that trend means that the sample is actually being affected by the power. So we need to be very careful with that, especially with single cells. So here we have a mixture of labeled and unlabeled cells. So if we tune the laser to amide1 in unlabeled bacteria in 1655, we can image the cells, we can highlight the cells that are unlabeled. And if we tune the laser to 1617, we can reveal the cells that are labeled with 13C. So this is a very standard procedure that we are constantly using in the lab, especially when we're working with antibiotic resistance. So the cells are usually, we grow them in 12C, in regular glucose, and then we treat them with antibiotics, and then we grow the cells after this in 13C. So if we can see the 13C shift, that means the organism is still active. So this is a very standard procedure, and now we can do this at single cell level, which wasn't possible with FTIR before. And we can see that there's variations from cell to cell, which is something that we're very excited to do. So now in the second study, we use OPTIR spectroscopy to monitor the production of PHB in Bacillus species. So PHB is a bioplastic, it's a plastic produced by bacteria. It stands for polyhydroxybutyrate. In this study, we used 14 Bacillus strains. 8 out of those 14 strains, they're PHB producers, and 6 of them do not produce PHB. We use OPTIR and FTIR to analyze book populations and OPTIR to analyze single cells. We did not use Raman. Actually, we did, but the samples were fluorescent, so we did not. So here we have the infrared signatures from a PHB non-producing strain. So it's like very similar to what we get with cells and serum, amide1, amide2, carbohydrates. And here we have the infrared signatures from a strain that produces PHB. So the additional peaks that we see are associated to PHB. Here is the infrared signatures from commercial PHB. They don't match 100% because the PHB, when you extract the PHB from the bacteria, you change the crystallinity and the concentration. So that's why they don't match 100%. And here we have the spores plots, the PCA results for the FTIR results from both populations. So we can clearly see the formation of two different groups. So one of the groups on the negative side of PC1, we have the strains that do not produce PHB, and on the positive side, we can see the strains that produce PHB. In here we have, similar to the OPTIR spectroscopy from book populations, once again, on the negative side of PC1, we can see the strains that do not produce PHB, and on the positive side, we can see the strains that produce PHB, but this time the reproducibility of the data is quite low. So the reproducibility is the distance between the spores from the same strain. So in the FTIR results, you guys can see that they cluster all together, and here on the OPTIR from book populations, they're kind of spread. And that indicates the ability of OPTIR to probe variations in PHB concentration across the book population. So in different spots of the book population, there are different amounts of PHB, which is kind of expected. And this is for OPTIR spectroscopy from single cells. So each one of those spores corresponds to one single bacterial. Once again, on the negative side, we have the cells that do not produce PHB. On the positive side, the cells that produce PHB. And if we plot the loadings associated to the first principal components in all three situations, they look very similar. And if we plot and compare them to the PHB, we can see that the discrimination patterns are due to the variations on PHB. So PC1 is dominated by this characteristic. And now, let's give a close inspection on this strain here, Bacillus megaterium, so the blue square. So in the FTIR results, all of the spores were clustered on the cluster of PHB producing strains. On the OPTIR from book populations, once again, all these spores were clustered on the PHB producing cluster, but this time, as I say, the reproducibility is quite low. But now, at single cell, there is something very interesting. So here, some of the spores, so some of the cells that we probed, they were actually clustered on the cluster of non-producing cells, strains that do not produce PHB, while some of the other spores were clustered on some of the other cells were clustered on the group that produce PHB. So basically, we're in different cells. We're probing cells that are producing PHB, the cells that are not producing PHB. So this shows the ability of OPTIR to probe cell-to-cell variants, which is something that we don't see with FTIR spectroscopy when we analyze book populations. I think Mustafa recognized this figure, right? This is from his PhD 22 years ago.
Thank you.
I didn't say anything. I was 9 years old. So you can use infrared spectroscopy to quantify PHB. So in this situation, we need to construct a calibration curve. So in the x-axis, we measure the PHB content using another technique such as GCMS. And on the y-axis, we use the spectral signatures, and then we construct the calibration curve. We can extrapolate the analysis, the data that I showed before for single-cell analysis to this type of analysis. So we can measure the PHB amount at single-cell level. This is something very, very important. Last but not least, we use OPTIR spectroscopy for bacterial typing. So in this study, we used 12 bacterial isolates. So these samples were collected from children with bloodstream infections, so sepsis. And we use FTIR, OPTIR, and Raman from book populations and OPTIR spectroscopy to analyze single cells. So here we have the infrared signatures acquired with FTIR, OPTIR book, and OPTIR from single cells, the spectra and the secondary derivatives. It is very, very similar. So it shows the high reproducibility comparing the results with the OPTIR with the standard FTIR spectroscopy. Here we have the PCDFA spores plot from the three different situations. We can clearly see two groups of gram-negative and gram-positive bacteria. These results were accepted for publication yesterday, so I'm very happy. And these are the Raman results. So here in the Raman, so when we compare the Raman signature from the different strains, we can see some peaks associated to pigments like carotenoids and cytochromes. And on the PCA spores plot, so basically on the positive side, of the PC1, we can see the strains that are producing pigments, and on the negative side, we see the strains that are not producing pigments. So we don't see this with infrared spectroscopy, what we can see with Raman because of the resonance Raman effect. And if we look to the second principal component, we can discriminate the gram-negative from gram-positive, which is similar to what we got using infrared signatures. So for example, if we have used 785 as probe instead of 532, we would not see the peaks associated to pigment. So we wouldn't have the resonance Raman effect. And we will probably see a similar pattern compared to what we saw with the infrared signatures. So if you guys, wanna follow me on Twitter, I'm the only Cassio, literally the only Cassio. And thank you all for your attention.
Any time for questions?
It was very quick. Yeah. Yeah.
And I was wondering
Yeah, well, it is metabolized because the cells they break it down and then they use those carbon to to proliferate. So it is the metabolize. No, we haven't done that. No, we have not. But Roy has done a lot of work using LCMS, so mass spectrometry to monitor this. Yes.
I've got one. Hi. Have you considered fusing the data and actually processing the two data sets together, sort of data fusion concept, and are the results any different, better?
Like Raman and infrared?
Yes, Raman and infrared.
So usually people do that when they don't have good discrimination using them separately, right? And we got some down.
Any other questions? No. We've got a last speaker now. Here we go. Professor Dr. Christoph Graf from my PhD.
Yeah, hi everyone. It's a pleasure to be here and to see everyone again in person. Yeah, 2 years ago. And my name is Christoph Graf. Some of you might know me. I'm working in Jena at the Leibniz Institute of Photonic Technology. There are also other people from Jena around here. So I started working with infrared spectroscopy as a PhD student in 1994. And well, in 2001, I was the first user of an infrared imaging system with a focal plane array detector in Dresden. And then I moved to Jena in 2008. In 2009, I was able to buy a focal FPA-based infrared system from Agilent. And I was in contact the first time with Mustafa. And now he convinced us in 2020 to, well, buy an OPTIR system. So since December 2020, I'm using the system. Unfortunately, I haven't published any data. I have so many other projects, but there are several present publications in the pipeline. Okay, title of my presentation today: Optical photothermal infrared spectroscopic applications for microplastic cells and tissue studies, comparison with FTIR and Raman spectroscopy. So I start with my personal experience in Raman spectroscopy. Then we did some work with bacteria, microplastic cells, and tissue. So I mentioned since 1994, Raman spectroscopy of biomolecules in Berlin. Then here the first infrared imaging instruments in Dresden from Brucker, and then the one in Jena. This is an extension for a macro chamber, comparable to here. You can move the SPA here also to the other chamber. And then in 2020, OPTIR. Here in our instrument, the Raman option was moved to the right side to the left side. This saves some mirrors and you get slightly better sensitivity. Yeah, our system is the first one with the 785, or was the first one with the 785 probe laser. Okay, we have seen in the previous presentation OPTIR spectra of bacteria and also FTIR spectra. So in our model system is the bacteria Bacillus pumilus and Escherichia coli from Ute Neugebauer's lab. And you see that can be nicely discriminated based on these bands due to the cell wall. And similar changes are also evident in the OPTIR spectra, which has been collected in reflection. Here, these FTIR data have been collected in transmission. But of course, if you look carefully, here are some bacteria, single bacteria isolated, and of course, it would be interesting to study them OPTIR imaging that can nicely be resolved. And if you collect different spectra from different bacteria, they're all very similar, but surprisingly here, these are, I think, two bacteria next to each other. If you look closely, there are some small changes in the shoulder of the amide band. Well, I don't know why, but it's interesting to see that there are some variations which can be detected with the OPTIR system. Yeah, then microplastic gives very nice infrared and also Raman spectra. So, and we studied here polyethylene, polypropylene, PVC, PET, polycarbonate, polystyrene, silicon, PLA, and PMMA. We collected OPTIR spectra here, this red curve, and FTIR spectra in attenuated total reflection mode, because the particles and the samples were quite large, and you cannot get good FTIR spectra in transmission if the particles and the samples are too thick. Now, of course, if you collect spectra in ATR mode, you have to correct the wavelength dependent penetration by this formula, and then OPTIR and FTIR, they coincide quite nicely. There are sometimes a few variations in the relative band intensities, and I will come back to the agreement in a few seconds. Okay, but first take a look. The Raman option is the OPTIR spectra and the standalone Raman system. So we use the Raman system from Kaiser Optical System, which is in use since 2008, a very stable system, sensitive. And the Kaiser system also has a calibration device. That means the spectra with the Raman system from Kaiser Optical System, they are calibrated, and of course, the same calibration were also required for the Raman option of the OPTIR system, because we observed here this blue experimental spectrum from the white light source, and the black spectrum is the true spectrum of the white light source. And if you calculate the ratio, you get a correction factor. So, and if you apply the correction factor to the experimental OPTIR spectra, then you also get a good agreement between both spectra, standalone Raman system, and the Raman option of OPTIR for these 9 plastic samples. Yeah, and we not only want to qualitatively compare the spectra, we also compared the spectra quantitatively. We calculated here the SID quality index. That's the product squared divided by this product And then if you compare the hit quality index of the infrared system with the OPTIR spectra, you get the highest agreement along the diagonal, and the off-diagonal data are close to zero. Ideally, for example, here, the agreement between polycarbonate and polyethylene gives a hit quality index of 0. The same with Raman, also the highest values along the diagonal. And also here, for example, polycarbonate with polyethylene gives then also a value slightly higher. And then Mustafa already inspired us that a two-dimensional identification approach is possible. So here are the correct identifications along the diagonals, and then all the other values are here close to 0 or between 0.2, with one exception. Here this PVC overlaps significantly with the PET spectrum, and the reason is there are carbonyl bands in PVC, and this is unexpected because there is no carbonyl in the structure of PVC. However, PVC contains plasticizer to make it elastic, so and this is why the carbonyl band is present. So here again is PVC, that's the structure, there's a chloride band, and then the rest is hydrocarbon, and still we have this strong band, which is unexpected. Yeah, and in this case, you can use the Raman hit quality index, which is quite low, 0.13 between PVC and PET. In this way, the 2D identification is more advantageous. However, also the Raman spectra can be affected by some distortions. For example, if you have pigmented plastic or particles, they can give a high autofluorescence background, or you get resonance enhanced contributions from the pigment. For example, very famous, the blue pigment, the phthalocyanoline, they are used as a very popular dye. Okay, next topic. OPTIR microscopy imaging of single-dried mesenchymal stem cell. This data were collected by Mustafa remotely when, I don't know, he had some spare time at home. And so he found this interesting cell. This is a mesenchymal stem cell, quite large cell. I published a paper, I think 2007, 2008. I still have the cells in the lab for some tests, and it was good to keep them. So this is the region of interest, and you see here the cell body. You see here green, a large accumulation of glycogen. This was typical for non-stimulated mesenchymal stem cells, who they maybe accumulated glycogen to have energy, and once they are stimulated, they can produce matrix. Okay, what else do we see? We see here a cell nucleus, and even we see the nucleoli, and there is this red membrane, and this red delineation zone is around 200 nanometer. That means even with the red laser, we get a similar resolution than with a green probe laser. Yeah, and this membrane, this delineation zone, has a very clear infrared spectral signature. So, and then we were we saw here Peter Gardner's paper about the work of OPTIR of cells in aqueous buffer. So I had a guest student. I said, here, let's try similar things, and it works quite well. So here we use the THP1 cell line, monocytic leukemia cell line. Cells were grown and then pipetted between calcium fluoride slide, and we found it was necessary to use very thin calcium fluoride slides, 200 nanometer. Otherwise, the contrast was very poor in the, in the microscope, and so we couldn't find the same. And of course, not only the contrast was poor, also the signal intensity was quite good. So we used also a sandwich pipetted with 2 microliter cell solution between on the first slide, and then covered it with the second slide. And then we collected data in transmission mode, and of course the water subtraction. So here under the AMAT1 band, there might be some spectral contribution with water, but we see a nice cell spectrum, and also we could resolve lipid droplets, and also some lipid droplets are as small as 670 diameter. Yeah, and then the preliminary measurements were finished, and we tried to use OPTIR also for classification of different cells. So students were very busy to collect almost 8,000 spectra. That means 5 spectra per cell, and then altogether approximately 260 cells for these HeLa cells, cervical carcinoma, this hyper-RG cell, liver cell, Raji cell, it's a lymphoma cell line, THP1, HL60, and Yogurt cell. These are all leukemia cells. Data were collected on 10 days, and on every day we collected, or students collected data from these 6 cells. And then classification was achieved by partially square linear discriminant analysis. And here in the model 1, the cancer cells were nicely discriminated. In model 2, the leukemia cells were well also discriminated with some more overlap. And then the model 3 was trained to distinguish the leukemia versus the non-leukemia cells. Okay, in PLS LDA, you also get information about the loadings. And so each loading has a certain signature, which is different for the 3 models. Well, we have here some effect, which is probably water vapor. As recommended by Peter, we will also use a PEP pen to seal the sample chamber, because I think in this simple sandwich configuration, some water vapor might evaporate, and then there are fluctuations in water vapor. But nevertheless, here are the tables with sensitivity, specificity, and accuracy, which are more than 90% for model 1, the cancer cells, which are around 82% for the leukemia cells, which show more overlap, which is again approximately 90% for the cancer versus leukemia cells. Okay, we have also projects where we study tissue sections, and one of the first OPTIR images from tissue was here from the bladder tissue section. This was 400 by 400 micron, or here 1000 by 1000 micron. There are a few dark regions, and this is and the reason is that the OPTIR system is very confocal, and you have to focus both infrared pump laser and the visible probe lasers within 1 or 2 micron. And if the tissue section is slightly, well, rough, then not the full tissue section is in focus. So spectra are very nice for typical bladder tissue, and of course here from the lipids you get almost a pure lipid spectrum. So, and then a second set of samples were collected from spleen tissue, and this was also one of the first work I studied with the OPTIR system with a focal plane array detector in 2005. And the function of spleen is first to filter out well, old red blood cells. So, and then you have these slightly darker regions which filter out these and accumulate and degrade the red blood cells. And then it's also part of the immune system. That means there are red pulpa, these are these follicles, and they contain B and T lymphocytes. Here they have been stained by immunohistochemistry. There is a darker ring inside and a lighter ring outside, and then, well, a zone, a bright zone in the center. And some follicles are completely bright. And of course, here this in the macro chamber, the resolution was quite high, but sufficient to resolve the follicles within the red pulpa in blue and cyan. And then from some follicles, we collected high resolution FDR images with 4x4 micrometer pixel size. And here these cracks, well, had to be removed. And then we clustered the remaining spectra, then resolved an outer ring and an inner ring of these B and T lymphocytes. And one of the features is here the elevated nucleic acid bands in these lymphocyte spectra. Yeah, now we studied spleen tissue from mouse. A parallel section was also studied by multifocal imaging, cast-to-full fluorescence, and second-order generation. And here the green area corresponds to the margin of the follicle, and also some contrast is evident for the cast channel for the red pulpa. In images of OPTIR, you not only get the OPTIR channel, but also a light gathering channel. And this already gives some contrast, for example, to distinguish red pulpa from the follicles. Yeah, and the follicles, when we collected here at 1086 and 1665, so this is a typical phosphate band, this is an amide band. So we get we observed also in the follicles a high nucleic acid content. And of course, in red pulpa, another spectrum, and then at the follicle margin, slightly reduced nucleic acid content. Talk about time with a 1 micron pixel size and a speed of 1000 micron per second, collecting a single image 1500 times 1500 micrometer is something like 25 minutes. A small region of interest was collected with 100 nanometer per pixel, and this was much faster, smaller region of interest, and you could resolve single cell nuclei. Here, these some of these spots, or most of these spots, have a size of 4 micron, and that's the size of a typical cell nuclei. Also from these spots, we got also maximum intensities of nucleic acid. And I think several years ago there was a debate: why don't we see the cell nucleus in FTIR spectra. And I think it was several reasons. First of all, the cell nucleus was not hydrated and dehydrated. Cell nucleus gives lower nucleic acid bands. And but of course, these tissue sections were dried, and using the indirect detection process enabled to collect these spectra from the cell nuclei in tissue section. Yeah, altogether we had 10, we studied 10 tissue sections from wild type and telomerase knockout mice. So when you knock out the enzyme telomerase, this affects the aging of the mouse, and we wanted to study some age-related effects. However, when we compare the spectra of the cell nuclei, there was almost no difference, and we continue this kind of research because our clinical partner wants to induce infections, and then we will repeat these experiments with infected mouse and study organs like spleen, and we also had liver and kidneys tissue sections. So there's a lot of work in front of us. As summarize, the benefits of OPTIR for studies of bacterial, microplastic cells and tissue is, first of all, the data are signal independent and can be collected in reflection mode. Yeah, thin samples can also be studied in transmission mode, like the single cell. Images can be collected rapidly at selected wave numbers. And of course, you can collect full spectra, infrared and Raman spectra at selected position. You can also collect, of course, a hyperspectral data set. That means a full spectrum at a certain grid. But of course, it's more time consuming than collecting an image at selected wave numbers. Resolutions in the sub-micrometer range, line shapes are not affected. No me scattering, as Mustafa already told. You can use a wide choice of substrate. Calcium fluoride is possible. Mirror IR. You can use metal, pure metals like gold coated. And someone visited last week my lab, and she prefers aluminum coated substrates. And even unexpected glass slides can be used, but then the sample should be a little bit thicker. The data are less water sensitive due to the optical detection principle, and our system was also upgraded to polarized option, so you can collect also polarized infrared spectra. The QCL is by nature, due to the signal generation, linear polarized, so you just have to include an analyzer in a certain position in the detection path, and then you can collect polarized infrared spectra. That's all for today, and I hope that maybe there are a few minutes for questions.
Running ahead of schedule. I think my 10 minutes that I missed has been carried on, so I understand my opportunity, but we've got at least 10 minutes, if not longer. Any questions, please?
Maybe I have a question. I very much enjoyed your work with different cell lines, very systematic, really nice foundation for the future to come. I'm just wondering, how come you had 3 models being built in the classifications and testing that? You have measured 6 or 8 different cell lines, right?
6 cell lines, yeah.
And you were comparing, you just built 3 models, 3 classifiers.
Yeah, we also
because the
yeah.
others are too different, or the others were running separate experiments, or what was the reason?
Yeah, so of course, we also took a look to build a model, try to separate all 6 cell lines in a single model.
Yeah.
However, this was not very successful because of the overlap or the spectral similarity of the leukemia cells. It was evident that the accuracy was in the 80% range.
Okay.
And so, and then we tried to focus on the separation of, for example, leukemia versus non-leukemia cell, and also we were surprised that we could distinguish the different cancer cell lines very well. So the HeLa cells, the HEPA cell, and the lymphoma cells. So this was in the range of 95%. So really maybe the selection of the leukemia cell lines was not the best choice. If we, for example, selected 6 cancer cell lines, well, in the past we also studied this pancreatic cell lines, this MIR-PACA2, and yeah, and of course we have several cell lines established in our lab, but at this time when we performed this experiment, these 6 cell lines were available, and yeah, and this was so both students, they were not very experienced in infrared spectroscopy of single cells, so I trained them, but after well few days they were able to really collect good data, and yeah, so and of course manuscript is under preparation, and I hope to publish it in the next months.
So, thank you.
Christopher, lovely presentation as always. I'm also interested in the cell line work that you did. If I understood it correctly, you took 5 spectra from each cell.
Yes.
Given the heterogeneity in the spectra, the spatial heterogeneity, how did you select where those 5 spectra came from? Because, you know, if you hit the nucleus or the cytoplasm or a lipid droplet suddenly, within one cell, you can get huge differences in the spectrum. So how did you take 5 and average, or yes, how did that work?
Yeah, so the cells had a size between 10 and 20 micrometer diameter, and so and they selected 5 arbitrarily positioned, maybe 1 in the center, and then 4 at the margin, and then the 5 spectra were averaged to compensate the variations that come from the different cell positions. So sometimes you hit the nucleus, and sometimes maybe the lipid droplets, and so the averaging should compensate this the intracellular variations. Of course, you could also, if you automate the whole process of cell identification and collection of spectra, and then move from one cell to the other. Our dream is to automate the process, the data acquisition process, and then you can easily collect more than 5, maybe 10 spectra per cell, and then you have even a better average.
I would comment on that. I think it's one of the advantages, but also disadvantages with the really high resolution. You're sort of exposed to the heterogeneity when you want an average in the cell. And one of the features of the mirror instrument is when we bring the instrument from the bottom, is the flexibility of the objective on the top collection, probe collection. So that you could use a low mag, low NA objective with maybe a couple of micron pixel resolution or spatial resolution. And then maybe 5 of those would do a better job generating a representative cell spectrum than 5 spectra or ultra high resolution spot size. Any question?
In one of your slides, it seems you have compared OPTIR Raman, the Raman data collected from OPTIR machine, Raman data collected by Raman machine.
Yeah, from the microplastic, I could
Yes,
compare the OPTIR spectra with the FTIR spectrum, and then the Raman spectrum collected with a standalone Raman system, and the Raman spectrum with these Raman option of the OPTIR. Yeah.
Did you see any bandwidth changes?
No.
Nothing.
Well, of course, we have to calibrate not only the intensity axis of the Raman system, you have to calibrate also the wave number axis of the Raman system. Yeah? And I used the Raman spectrum of polystyrene for the wave number calibration. This is also a recommended procedure when we discussed the calibration issue of Raman spectroscopy and the cost-action Raman for clinics. Polystyrene was one of the test samples which were sent around in a round robin trial, and polystyrene was one of the plastic spectra. So I used the plastic spectrum and the known bands, the Raman bands of the polystyrene to calibrate every spectrum from microplastics. And then I got very high accuracy in the wave number axis, and of course the good accuracy and in the intensity axis was achieved by the wavelength calibration, the intensity calibration with this white light accessory from Kaiser Optical System.
But there was no bandwidth difference.
Well, the spectral resolution was comparable with the Raman option of the OPTIR system. So there are 3 different ratings: 300, 600, and 1,200 lines per millimeter. And I used the middle one, the 600 lines per millimeter, and the spectral resolution was in the range of 5 to 6 wave numbers, which is similar to the spectral resolution of the standalone Raman system from Kaiser.
I can't think of anything else for you to say at this point other than to thank all of our speakers again. Thank you for your time.